बंद कर दो ना, आवाज आ रहा है बाहर से.
Delegates are mostly back.
Welcome back from the break, everyone. I hope you all had a refreshing pause. Now, up next, I'll call Dr. Nikhil Patkar, who will introduce us to the error-corrected sequencing, a breakthrough method that enhances the accuracy of sequencing by correcting errors, offering more reliable insights for research and clinical applications.
Thank you. So, hi, good morning everyone. So I was supposed to take a talk in the morning, but I had to postpone it for some reason. So I'm going to talk on error-corrected sequencing in AML. This is my most favorite topic in the world because I do extensive research on this. I develop methods. I do, this essentially is the crux of my research career. So most of my research is on AML. So a little bit of background on AML. As we all know, AML arises from a clonal expansion of myeloid blasts, and other than 1517 has heterogeneous biology and outcomes. So we all know that patients with intermediate cytogenetic risk have heterogeneous outcome, and understanding of AML biology has greatly been linked to improvements in technology. So 1970s, this was entirely a morphologist game, wherein we had the Fab classification and everything was classified based on how it looked under the slide and what how the cytochemistry of it was like. So we had the famous FAB classification, which was there, put forth in 78, 79, was practiced all throughout the 80s. But in the late sometime around that time, you had the late 90s or something, or in the 90s we had the EGIL classification, which said that immunophenotype is also important, and based on that they used to classify leukemias. But around about that time we also had microarrays for human genes genome, as we saw yesterday, and at that point this gene expression profiling was a very, very intensely favored topic amongst researchers. All the way till the early 2000s this was pursued. It although didn't really come anywhere close for clinical interpretation, but that was it. After this, the genome of this disease AML completely exploded with the genomics era, with the first cancer that was sequenced was that of an AML patient, an AML M1 patient, through which we identified DNMT3A, IDH1, IDH2 mutations. Subsequently, large cohorts did the TCGA, Atlas, and numerous cancers got sequenced. Today, this has translated into the clinic. We use this as a way of diagnosing diseases, as a way of risk stratifying AML and such. So AML, as compared to other malignancies, is mutationally less complex. All right. So there are approximately, if you do a whole genome sequencing, 8 to 10 mutations. But if you do this for a disease like melanoma, you will get more than 100 plus mutations, and it's quite bewildering. It just shows that this is a mutationally less complex disease. And even if you look at cytogenetics, even then there is more heterogeneity. Certain disorders, such as those with core binding factor leukemia, RUNX1, RUNX1, T1, CBFB, MYH11, they are mutationally less complex. That means they harbor less abnormalities. Even today, with using an extensive 135 gene panel, if you see which these patients can sometimes just have no mutation, it is patients with CBFB, MYH11, and more commonly patients with RUNX1, RUNX21. You will just not get anything. So this just highlights biology of disease. As opposed to that, if you look at intermediate risk AML, full of mutations. If you look at patients who have unfavorable disease like if they have monosomy 5, monosomy 7, sometimes those patients also may not have any mutation. So these discovery studies, we understood that, look, it's not just any gene. These genes tend to cluster along genes encoding for pathways. So genes encoding for spliceosomal genes, genes encoding for transcription factors, genes encoding for tumor suppressor, genes involved in DNA methylation, and that gave rise to this pathway-based medicine, systems biology approach, and we now just focus on those genes to make them as targeted panels. So if you use a large or even a small panel for AML, this is not large, this is just 50 gene panel. If you use a 50 gene panel for AML, you can identify a mutation in nearly 90% of our patients. And this is data that we published some time ago, and it shows that, FLT3 mutations are very common, NPM mutations are more commoner than RAS, than DNMT3A, WT1 and so on. So if you compare this with Western data, you will say, wo to bolta hai DNMT3A, TET2, ASXL1 is most common, but it is DNMT3A, TET2 at least are more common than this. So it does not mean Indian genome is different. It means that there is a referral bias that is there in our hospitals. In our patients, if you see the median age for almost all public hospitals, I can say that the median age where we treat AML is younger. Because a 60-year-old Indian person may not have the same fitness as that of a 60-year-old German. And so they will not take them up for intensive chemotherapy, and therefore these are underrepresented in our cohorts. And this is the genome what you would expect for a median age of this cohort was 37. So for a 30-something-year-old AML, this is what we see is common. So which brings us to the question, if you can get mutations in so many patients of AML, can you track them, and can this be a surrogate method for MRD detection in AML? Before you start doing MRD detection in AML, there are certain things that you must be aware of. That is, what is age-related clonal hematopoiesis, or CHIP. So CHIP was a phenomena that they identified in 2014. 3 seminal papers were published in the same year. One was from the CHIP cohort, one was from a cohort of patients with heart disease, and another was a cohort of exomes from patients with schizophrenia and other mental disorders. They didn't identify heart disease or schizophrenia causes, but what they did identify that as patients start to go older, although they are completely hematologically normal, they start developing mutations which have been described in hematological malignancies. Typically DNMT3A2, TET2, ASXL1, and now SRSF2 and IDH1 are also part of this, which gave it to the concept of how do you distinguish AML-related genetic abnormalities from clonal hematopoiesis type genetic abnormalities. So AML-related genetic abnormalities, for example, NPM1, occur later in the clonal ontogeny of AML. Whereas those mutations in clonal hematopoiesis are at least for adults, are the initiating or the founder events, and they occur at a VAF which is typically higher. If you give these patients intensive chemotherapy, alright, these mutations still persist. But the driver mutation, the other mutations that are there like NPM1 and so on, go away. In relapse, those mutations come back, but these persist throughout. The only way to get rid of these mutations if the patients undergo bone marrow transplantation. So this is evidence of what I'm saying. These are patients who are co-mutated for NPM1 and DNMT3A. This was what we published way back in 2018. And you can see here that these are same patients. Each color here corresponds to one patient. So see this orange fellow. This orange fellow's NPM1 mutation drops to 0.001 or somewhere around that thing. But his DNMT3A mutation is persistent. All right. So look at this patient. From high VAF, his mutation dropped to 1%, but his DNMT3A mutation persists. So this is what we mean by these originate in CHIP, and they are persistent in spite of intensive chemotherapy. And this is seminal work that was published from Peter Fox group at in Netherlands, and they showed that if you exclude these DNMT3A mutations and you can get those mutations at post-treatment time points, that means post-induction, post-consolidation, if they are there, these are surrogate markers for MRD, and they can be used for MRD detection. But is it so simple? Like you take a sample and sequence the hell out of it. Like if you sequence something at 100,000x, can you go up to 0.0001? Does that mean it's like that? So unfortunately, this is not so straightforward. All right, because if you look at a sequencer, this is any short read sequencer, Illumina hai, Iron Torrent hai, whatever. If you look at a IGV plot, you can see that suddenly there'll be a G somewhere, suddenly there'll be a T somewhere. These are all changes from the reference genome. These are actually errors of sequencing. So why do these errors arise? These errors arise because of multiple reasons. One, library preparation. When you start doing library preparation, you have to chemically shear the library. This is done by fragmentation process, typically enzyme related process. These itself induce errors during the library prep process. Two, when you do end repair, the repair processes may not be so intact so as to repair this change. 3, if you are using formalin fixed tissue or any old type of DNA, formalin fixed tissue and old type of DNA bring about chemically induced changes of C to T, which are called, I forgot what it's called, but these are chemically induced, deoxo 8G. They produce deoxo something change at that particular base, and these bases are very persistent there. If you look at a chemical genome, a formalin fixed genome or an old tissue, it will have much more of these C to T mutations. All right. So these are changes that are there in ancient DNA. Second thing is, if you do high amount of PCR too has a processivity and a proofreading there. You can use high proofreading enzymes, but even then there is an error involved. Right, and those PCR also will start introducing errors with each cycle. And how do you get rid, or how don't we see these errors in common clinical practice? So you must understand how variant calling is done. So far we have not described actually what goes on into variant calling. All variant calling works on Bayesian statistics. That means it takes a base and then it takes multiple calls that are there, and then based upon the reference genome, it makes a statistical probability that what is the likelihood of this being a true base, true thing as compared to a false artifact. And almost all algorithms and methods that are there will have a cutoff there that hovers between about 1 to 3% or 3 to 5%, depending on how your assay is set up, but typically in the 3% ballpark. Which is why you cannot do MRD detection without, just by sequencing the hell out of it. It will not detect low level abnormalities. And this is why most clinical assays in any lab, whatever method you use, will have a limit of detection of 3 to 5%. So what can you do? You can do statistical modeling. So statistical modeling that is used to get rid of these errors is called as position specific error modeling. So that means that you take a sample which is a well characterized reference genome. So these are typically cell lines. So do you know that Cosmic has a cell line encyclopedia? All right. Similarly, there is a reference HapMap cell line that is available. So these are cell lines in which we have characterized the genome extensively. Now you take these reference genomes and you sequence them multiple times, and then at every base you count what were the number of bases that were called. So you will have a frequency that is there. So if the reference is C, you will have out of 3,000, or like say 10,000 C's, there were 50 T's, and then there were 60 A's, and there were so and so that are there. And then you come across, and then based on this, you can say what is the change from the reference to C, G, T, or A, depending on what excluding what the reference base was. And then if those are there, you now have the incidence of those changes at that base, based on which you can make a statistical probability, because you have you have an event, you have its mean, you have standard deviation, from which you can use chi-square statistics or whatever mathematical modeling to say that this is a statistical probability of an change. You make a matrix of this, and this matrix now gets incorporated into your VCF file. So based on the chromosome reference start, alt, stop coordinates that are there, if a C changes to a T, we can have a statistical modeling to say that, look, what is the statistical likelihood of this being this. All right. So this is a way of doing it. It is. It was what was done. If you ask me what Peter Folk did, they did not do error corrected sequencing. They did this position specific error modeling. All right. But there is another way that is done to prevent errors, and that is a wet lab method. And that's called, the wet lab method is called single strand consensus sequencing using unique molecular indices. So one of the methods to do this was safe-seq, where you take DNA and you do a limited cycle PCR. That means you just do 5 cycles of PCR of this. So you not only enrich your target, but while doing the PCR, you introduce UMIs. So what are UMIs? UMIs are actually oligonucleotides where the bases are truly random. So you'll have a stretch of 8 nucleotides which are, if you want to get them synthesized, you say N. Like it can be 50 Ns, 30 Ns, 20 Ns, 10 Ns. And those will be random bases. What does that mean? That means that every oligonucleotide can bind only to one strand of DNA. Since one oligonucleotide binds to one strand of DNA and the UMI is truly random, every DNA then gets tagged with a unique molecule. It becomes like a fingerprint. Okay, it becomes like a fingerprint, and therefore you can do PCR amplification and every molecule gets tagged uniquely. But the problem here is you are still doing PCR, and in those first 5 cycles you can still introduce sequencing errors. And anybody knows that a 5 cycle PCR is not at all fun to deal with. It is going to be difficult. It's technically difficult, especially because the genome has varying GC content. All right, so one is that. The second is single molecular inversion probe. So I think I described to these to you in library prep what are SMIPs. These SMIPs basically, when they bind, here there is no PCR cycling here. The SMIPs come and bind, they introduce a UMI and they make a circle, and then when you do rolling circle amplification, you get a library. All right, so we will discuss on how errors thing is done. So this is single stranded UMI. Why single stranded? Because you are taking only one strand of the DNA, the other still may have errors. Next is duplex sequencing. So duplex sequencing is like the next level of UMI based sequencing. In duplex sequencing, it's not just one strand, but both the strands of DNA that have a UMI attached to them. All right, and the strand that is there on the upper strand, the alpha strand, the UMI, its complement is attached to the beta strand. So if you have a double stranded DNA, here you can see that the UMI that is here, its counterpart will be here. So if it's A, the UMI on the other side will be T. All right, just to give you an example, because A binds to T. So how does this error corrected sequencing work? Is that once you have incorporated the UMI, you have to amplify the library and do PCR. Now since this is AAAA, you do PCR, all the PCR copies will be AAA here. Okay, but what will this be? These are random errors that you introduced. So when you do bioinformatics, all of this gets collapsed together. And these errors go away. And true mutation, what you see here in purple and what you see here in blue, will be held together because they are present in all the reads. And all of those reads get collapsed together to form one read.
So won't the errors get incorporated into that UMI?
No. How errors are so UMI never gets incorporated by doing PCR. UMI is always or as far as possible is introduced using a PCR free approach.
Then how would you amplify subsequently?
So once you attach the UMI using a PCR free approach, subsequently PCR can come. But UMI, all the things that are there, it is the same consensus way. If all AAAs are there, you know that if there is a suddenly T in one UMI, it can be discarded or changed to an A and that read is incorporated. Okay. So there are ways. When you do this collapsing, there are ways in which you take this consensus. Errors in UMI are not that common. Errors in these kind of errors are common, and that you have to again tweak your variant calling to say what actually constitutes a consensus. So we use typically 70% of the reads should have that mutation to say that this is a consensus. You can make it more lenient, or you can make it more stringent depending on how many errors you are getting. वो बाद में लास्ट स्टेप में आता है, किसी भी स्टेप में वो बाद में आ सकता है, उसका कोई generally. So it depends on how your library prep is set. These methods like MIPS, the UMI is added first, and then the index comes. Here also UMI added first, index comes. In the Aloha seq that we have, the UMI is added first, late third round index is added. Alternatively, one index and UMI are added first, and in the final round an index is added. In case of if you are using those adapters by twist, or if you are using Y-shaped adapters, in that UMI and index are added at the same shot. Okay, so it depends on the library prep. It really doesn't matter. The general caveat here is like this. Okay, so for duplex sequencing also, alpha and beta strand. So if one strand is there but other strand is not there, it is still discarded, and only if they are there on both the strands, that is retained. So can someone tell me what is the negative disadvantage, or what is the disadvantage of UMIs? From all of this that I have discussed, what is the disadvantage? So the disadvantage here is redundancy of sequencing, because there are so many PCR cycles that are taking place in any library prep. PCR cycles take place, but because there are PCR cycles take place, and you are collapsing all PCR duplicates into one read, it essentially means that almost 70%, 60%, or 50% of your reads are going to be thrown away or discarded. Typically this hovers around 70%. All right. So there is a lot of redundancy of sequencing. So just to give you an example, that in our this paper that we published, our median read depth was 25 million reads per sample. That for the entire Illumina V2 library is 25 million reads. Okay, whether it's for Minisec or whether it is for MySec. If you are going to do, this is an expensive affair. It is not well suited for desktop sequencers. The same thing is there for Ion Torrent. Ion Torrent 2 has, I believe, an error corrected sequencing assay that is there. But this disadvantage stays, that you can only put 3 or 4 samples on small sequencers. Right now we do it on a NextSeq where it is more affordable, but it still is kind of expensive. Now why do you want to do this? Anytime you want to detect alleles or anything post chemotherapy or post therapy which is there, you require such kind of approaches. So it is there for AML MRD. You can use it for ALL MRD if you're tracking mutations. You can use it for kinase domain sequencing if you are trying to do that, although that is not the best example. You need to do it for liquid biopsies where typically the mutational fractions are low in solid tumors. So these concepts are universal across the board. You can do it for then whatever applications where you can think of where rare allele detection is required. This is the approach to use. So so if you see using single stranded UMI technique, the typical limit detection is 0.1. If you use duplex sequencing, this 0.1 goes on to become 0.001%. All right. So those are the kind of limits that you are looking at, 0.001 or 0.001 depending on what you do. So do you always require to use UMIs when you want to do error corrected sequencing? And the answer is no. If you have complex indels, this is only true for short read sequences by synthesis wala chemistry. So if you're using Illumina or other things, you actually don't require UMI sequencing for things like NPM1. Here, NPM1, the most common way to detect the mutation was by qPCR. This is famous paper by Adam Ivy which showed this. We developed an NGS MRD assay wherein a single but the problem here is that every one of these mutations requires a separate type of real-time PCR. So which is crazy. Type A, type B, type D, all of them. These are most common ones, but there are more than 50 different mutations that are there. So see this, we developed a single UMI based You can do that. You can do that. What we did is use one assay for everything. So here, this is just one PCR, single PCR assay, no UMIs or anything for NPM1, and you could detect whole lot of any known or unknown. See, look at this. One is a novel NPM1 mutation, but all of these we could track using this assay. Now, if you see the sensitivity of the assay as compared to real-time PCR, NGS-based MRD is actually far more sensitive. So this is well known across the field now. Now, this. How is UMI-based monitoring done for blood cancers? So it had been shown by Eric Dunkovich for post-transplant monitoring in MDS. It had been shown for pre-transplant monitoring using patient-specific approaches by Felicitas Thole and Michael Hauser. It was also shown by Chris Hurigan. So at that time, we had several NPM1 these assays in our lab. One was the MIPS approach, which introduces this. You can do MIPS and introduce library. You can do PCR and try to introduce the UMI by PCR, also doing limited target enrichment. And by Narasimha approach, here advantages without both these methods, advantages you without doing the first PCR, you can introduce the UMI. So the key here is before the PCR, can we introduce the UMI? Okay, so we wanted to ask a question: how does panel-based error-corrected sequencing compare to flow cytometry, and what is the outcome of these patients? So we developed a 34-gene hotspot panel for these MRD mutations, captured the genome using SNPs, amplified them, and as you can see here, all the reads that originate from one UMI will have the same info. If those mutations, if random mutations are there, they get collapsed and they form one read. All the reads that then map to the same region of the genome will get collected together, and these form consensus reads. Now we have moved away from this approach, and we have another approach called Aloha-seq. We have patented this, and we are starting a large cohort of AML to test for this particular mutation with a clinical question in mind. So for FLT3 mutations, again, we developed a one-step PCR assay, and you can see here that you can detect mutations. Generally, if they are indels, you can go up to 0.03, and if they are these SNVs, you can go up to 0.05. If you don't use error-corrected sequencing for FLT3, we could go as low as 0.02. So you can see triplicates here in a dilution at 0.002. You can go very, very sensitively for FLT3. So just like with any sequencing, there are some stringent criteria that you need to apply. So why do we need criteria at all? Because even now, even using all these approaches, there is still a background error. And if you use single stranded UMI sequencing, ऐसा नहीं कि तुम UMI use किया तो ये सब चला गया. Alright, you will get some level of background error, and you have to know when to take this and when to discard this. So if you see ELN recommendation criteria, they still recommend that minimum of 10 reads should be there for you to call that mutation. So the variant should have been detected at baseline. You also have to then incorporate an error model on top of this. So error model at that site should have a p-value of less than so and so. Minimum 10 reads should be there. Minimum 3 alternate variant reads for an indel. And the highest WAF should be taken as MRD. And if the highest WAF was in a variant associated with DTA, please ignore. So we took 200 patients in morphological remission. Most of them were intermediate risk. Small fraction received allogenic bone marrow transplant. Most received 3+7. You can see that as when you saw flow results, 44% were MRD positive, but 71% were NGS MRD positive. And for consolidation, 41% were NGS positive, 21% were flow positive. So this is in like most of the patients, you could get one mutation that could be tracked. And only out of those folks who had one mutation, only 3 patients had a DTA mutation, and we excluded them. All right. So this approach was applicable to 80% of our patients. Now with our new approach, we are finding more than, I think some Aarti did the calculation, at least 90% Aarti. So around 90% or slightly more than 90% we can apply this. The big improvement here is that we were not sequencing CE-BPA. Now we are sequencing CE-BPA also, as we found a way to deal with CE-BPA. And you can see here that, that DNMT3A mutations have a higher VAF, signaling pathway mutations, all of these can present at varying different VAFs. But generally, the median VAF for our approach was 0.76%. So this was the VAF at which we could detect, was the median VAF at which we detected MRD. And those patients, of course, who are MRD positive have a worse outcome as compared to patients who are MRD negative, and both for overall survival, relapse-free survival, as well as cumulative incidence of relapse. And maximum benefit was there in your intermediate risk category, and in favorable risk, there was a tendency, but it was, number of patients were not more. Hopefully we can address this. So 0.05 for SNVs, 0.03 for indels. If you looked at kinetics, patients who were persistently MRD positive had a worse outcome, and patients who were persistently MRD negative had the most favorable outcome. Yes.
And in comparison with flow MRD was also at that cutoff, sir.
Flow MRD, any MRD is positive. Any MRD. Because flow, AML, you cannot apply that cutoff. Any MRD we take as positive. So patients with persistent, like MRD negative, had better outcome as compared to patients who subsequently, where PI was positive, PC became negative, and so on. So also we found that, some of the patients, you could see the change in mutational profiles. So for example, new KIT appeared, some NPM1 mutation appeared, one patient PTPN11 mutation appeared, and so on. So this is an important plot where we show that if we compare the results between flow and NGS, patients who are negative by both modalities had an excellent outcome. Patients who are positive by both modalities had a really inferior outcome. And any method of positivity is actually bad. So this particular recommendation has been used by the ELN. They use our paper, and this is this and Peter Fox paper to make this criteria, which says that any method of positivity should be looked at badly. So we found that why do patients persist? Amongst biology, if you patients are RUNX1 positive, they are more likely to be MRD positive. We know RUNX1 is a horrible marker for predicting MRD. And same thing we see in Cox multivariate analysis. But we also see that flow, that both these flow and NGS MRD are independent predictors of outcome. We still, cases in which we could not do the analysis, most of them include ASXL2 and so on. You can also do MRD detection using RNA sequencing. You can, especially when you identify fusions which are novel. So for example, KMT2A fusions, they can be anything, NUP98 fusions, they can change from patient to patient. So Chris Hurigan first demonstrated a PCR based approach to detect these fusions. Using our Narasimha approach also, we showed that you can detect KMT2A, MLL-T3. Remember, we are only sequencing KMT2A, not MLL-T3, and you can detect these mutations at least till 0.1%. This is on a MySeq. If you use another sequencer, it can be much more sensitive. Lastly, there are also improvements in error modeling to improve these low VAF calls. We use an approach here, which instead of position-based approach uses a contextual-based error modeling and a complicated estimation called VBull estimation. And we have incorporated this into our pipelines now for more accurate error modeling post this thing. MRD detection. So,
Base is going to lower your detection limit further down. We'll be able to start calling cases positive.
The whole trick here is that this helps you distinguish background error more efficiently as compared to the position-based error modeling, context-based error modeling. So in that there is an improvement, and therefore you can, in the same number of reads, you can be more confident. Or another way to look at it is we can detect more from less. So sequencing based MRD is it has the main modality of MRD assessment. Combination on flow and NGS was the best. Now we are trying to answer whether leukemic stem cell MRD and NGS MRD, how do they compare. So this will be, hopefully we should have this publication out by the end of this year. With that, I'd like to say thank you to whole lot of people have worked on this. Thank you. Thank you. So why UMIs are not required in NPM1? So number one, what are the type of errors that are made by short read sequences? In Illumina short read sequences, typically these are base substitution errors. Okay, or you can have 1 or 2 base indels. So where do these come? 1 or 2 base indels, typically in SEPA. 1 base pair indel or 2 base pair, 1 or 3 base pair indels, typically in SEPA. 1 base pair, 2 base pair indels, all the time these errors are made. SNV, all the time these errors are made. But you will almost never get a 4 base pair indel in NPM1, never. Which gives you the advantage that you cannot use UMI and just do NPM1 based standard MRD testing to detect this. So you don't need to use UMIs. Okay? Thank you. What's in a new?
Thank you, sir, for that informative session. We were all eager to attend this, and it really clarified our understanding of error-corrected sequencing. Now we will continue with our session on HGVS nomenclature. For that, I invite Dr. Shadma to the stage. Let's stay engaged and make the most of these valuable learning opportunities.
Good morning, or is it afternoon? Okay. Yeah. So we will continue with the HGVS session and the saga of HGVS nomenclature. So ma'am had asked this question through Shakespeare, what's in a name? And if Shakespeare would have been with us in the morning session, now he would realize, I think it's got letters and stuff, and you have to give a nomenclature. So I'll move on to my first case. And me and my colleagues will be talking about cases here. And we have given those cases to you as well, and you would have analyzed. So you can cross-check the results that you have got. So this was a patient, suspected case of acute leukemia. Bone marrow morphology and immunophenotypic features are consistent with B-cell acute lymphoblastic leukemia. Cytogenetic analysis by Fish revealed tritetrasomy 21, trisomy 4, 10, 17 in 85 to 95% cells. And it revealed basically high hyperdiploidy. So BALL with high hyperdiploidy. And on NGS, we saw two mutations, PTPN11 and NRAS mutation, which are RAS pathway mutations, and they are known to be more commonly seen in BLL with high hyperdiploidy. So we already saw the nomenclature of PTPN11 in the morning session, how we do the HGVS nomenclature. And here, if you can note here that the change in the genomic variant is shown as A to T, and in the transcript variant is also A to T. However, the NRAS gene that we can see here, there is some sort of a discrepancy, C to T at the genomic level, but at the transcript variant, we have a G to A. So can anyone guess? And I think ma'am had also asked this question in her session. If anyone has any idea why this is happening, is it a typographical error or some other kind of an error? So what we did, as we did in our previous session also, we manually check it, and we went to the integrated genomic viewer of this, and we can see again the C2T change is there, and we take the sequence of that region. This is the sequence in this region, and we again check it on the ensemble. We all opened ensemble, and we saw this is exon 2 of NRAS. So here we are checking, we are mapping the sequence on this exon 2, and we see that we cannot find this sequence in exon 2 of NRAS. So again, why is this discrepancy there? Normally, ma'am also told everyone that if we put the sequence, we'll see the sequence there on the particular exon. So why is this happening? Any guesses? Yeah. So it's actually Gaurav sir's birthday today. So sir missed the meme. So I was looking. So before we go on, it's Gaurav sir's birthday, and we'll just wish him.
साल भर में सबसे प्यारा होता है एक दिन, 100 दुआएं दे रहा दिल तुमको आज के दिन. I wish you happy happy birthday, happy birthday, happy happy birthday to you. I wish you happy happy birthday,
Happy birthday, Gaurav sir.
happy birthday, happy happy birthday to you. I wish you happy happy birthday.
So why is this happening? Somebody answered, I think. Yeah, it's not because of the birthday, but it's because it's something to do with the reverse strand. So when you open ensemble here at this location, it also mentions whether the gene is on the reverse strand or it is on the forward strand. So this gene NRAS was showing to be there on the reverse strand. So what is the concept behind this is basically we know that DNA has two strands, and there is a forward strand and a reverse strand. The genes can be mathematically any gene, 50% of the genes will be on the forward strand or they will be on the reverse strand. So the genes that we covered earlier in the previous sessions, they were mostly the genes on the forward strand. So we did not have this issue. If we put the sequence there, you would have seen. But the reverse strand gene, the sequence will not be seen in the online, the ensemble databases. Basically, this is because these databases like DNA sequencing databases where the data is stored of the DNA sequence, these just have one strand stored to make it less cumbersome. Basically, you can just take the complementary, you can find out the complement of it by reverse complementing. So that's why for NRAS and other genes which are on the reverse strand, you have to reverse complement the sequence. So these genes on the reverse strand, they are having the same function. They are not anything different. They just their location is different. The rest of it will be the downstream process, the transcription, translation and all will just be normal. Only the position and their work is basically, they're like the anti-heroes of the day. So they just work in the opposite direction, but otherwise the rest of it will be the same. So these are the basically the reverse strand genes that we see in which are common in hematolymphoid malignancies, and like NRAS as in our case, and there are other common ones like KRAS, IDH1, IDH2, DNMT3A, U2A1, and when we collected this, we realized a number of hematolymphoid genes are on the reverse strand. So just like it doesn't matter where they are, their function is just the same. So the earlier we saw this PTPN11 and JAK2, they were forward strand genes, and if the sequencing data that we get in the 5' to 3' direction, so their sequence will be matching. But if the genes are on the reverse strand, the sequencing data that we are getting again will not be matching, so we have to reverse complement it and we have to then check the sequence. So the sequence that we had for NRAS that I took from IGV, I will now put it on a tool, reverse complement tool, which is in the bioinformatics.org website, and we put it there and we get the reverse complement of it. So you can see that this CCA has been reverse complement. CCA, so now it is GGT. So it's reverse as well as the complement of it. So it was starting from this direction. Now we see from the other direction because it is the reverse also. So CCA, GGT, and then there's TGT. So again ACA, and then CCA. And then this becomes GGT. So when I had copied earlier, I had copied the sequence on the other side of the mutation, right? C2T change. So now the mutation will be at this end of the change. Okay, our mutation earlier when I had copied here, it was from the here, this side. So this was the mutation, I had copied this. So the mutation was at this end. Now after I reverse complemented, it will be at this end. Because it is reverse. So now I put it in the ensemble, map it to the ensemble database, and there I see that this sequence is here, and this is the position where the change is happening. But this is showing again as a G because it is the complement of the change. So this is actually the change C, and that is getting converted to T, which is in the ensemble database because it is just for one strand, it is showing as G. And so we can now count here. So this was 115, 258, 798. So this is 798. After that, if you start counting, this will become this 8747. So this is the position 8747, where the C is getting converted to T, though on ensemble it is showing the complement of it, G. And the cDNA sequence that you had downloaded there, again it would be similar because it is based on the sense strand only. So the codon position 35, so this is the position 35 where the G is getting converted to A. So instead of a GGT, we now have a GAT, and if you have that codon list that you have with you, you can check what GGT is. So GGT was glycine, so now that is getting converted to GAT, which is aspartate. So this is the change. So this is what we have found out manually. What we learned in variant validator, that is how we normally report, and that is how you can go about it. But this is actually manually how we see this. So this was the question that we had put. So how many of you got this answer? You can check. So you would have got this answer by putting it on variant validator.
So basically, we just showed you manually, but for all practical purposes, we do it on variant validator and we get this result. We are just demonstrating the n depth of that thing.
So will this have any impact during primer design? Like the reverse complement, then how you have to reverse complement before ordering primers? Or?
So basically, when you are ordering, when you have to order exon 2. So in ensemble, you will be ordering the start coordinate and end coordinate of your NRAS gene, for example in this. So from there you can take that way. So that will sequence your event.
Okay, so no, because in ensemble it is already in the reverse thing.
Yes.
So when you are ordering primers, just go with that correctly. We need not do the reverse.
Yes, only thing when you order, you have to reverse the sequence. I mean, the start has to be first and the end has to be later, right, sir?
No, actually it does not matter.
Yes.
Because every DNA strand is double stranded, and if you look at the reverse strand that is there, even though you may order a reverse strand primer for the reverse strand, it will still bind to the complement because the complement is always going to be there. The issue arises when you are trying to look for cell-free DNA or issues where there may be single strands of DNA involved. There this consideration may come. The other thing is if you are trying to design primers for fusion, there it matters because you will end up otherwise sequencing ulta, like going ahead of the fusion, like BCR ABL1, you will end up sequencing in the direction of ABL1. So that sort of thing. So sorry, Shweta, that was not on.
So moving on, there's this case, 73-year male with pancytopenia under evaluation. Bone marrow showed dilute yet cellular bone marrow with trilineage hematopoiesis and no evidence of abnormal myeloid blasts or dyspoiesis. On NGS, we got a DNMT3A mutation at a VAF of 9.88%. So, DNMT3A mutation, as was also covered by Nikhil sir in the previous session. This seminal paper by Siddharth Jaiswal et al., which was one of the first studies, showed that DNMT3A mutation is one of the commonest mutations occurring in clonal hematopoiesis. And in this case, we also have pancytopenia. So this would be a case of clonal cytopenia of undetermined significance, CCUS. And there's another study, recent study, where they have shown that a single DNMT3A mutation confers a favorable outcome to the cases of clonal hematopoiesis. So how do we give the HGVS nomenclature for this common DNMT3A R882 mutation? So you would have put it in, we gave you in that questionnaire. So you would have put it, you can cross-check what this anti-hero result was. If you can check with your, how many of you got this result? Or you can just enter now. By the time I show you the manual results of this, you can again enter. Again I'll show you the results so you can cross-check. This chromosome 2, 254572 on, they've done it. Yeah, you can put it from Excel or you can just note it down from here. And by the time I show you the manual approach, then you can, by the time you can do it. So, how we go about it. This is the change, C to T. And we again have to take the sequence, because again this gene is on the reverse strand. So we take this sequence and we map it to, we first reverse complement it, and then we get the reverse complement sequence of this, and then we map it to ensemble, and there we know the sequence that is there. And this is the position where the change is happening. So this is a G. So actually the change would be the complement of it. So it is a C, and that is the change. C to T is happening, and this is the position 242. If we count here manually, it will come to 242. At the cDNA level, we have at this position 2645 position, there is a change from G to A. So CGC is becoming CAC. That is arginine at 882 position. This arginine is becoming histidine at 882. So now you can cross check with your results. So you would have got something like this on variant validator. Next we move on to the next case. So this is case number 6, a case of AML with NPM1 mutation, and there is an IDH1 R132C mutation. And we know IDH1 mutations can be therapeutically targeted. So this mutation is again important, and we normally see it in our reporting. So how do we do this reporting of, how do we do the HGVS nomenclature for this? If you can enter the sequences, if you have not done, and so this is also a reverse strand gene. So if you've entered, then we'll move on. So this is the thing you will get. So I'll show you manually how we got it. So there was a change at G2A, this position. We took the sequence of this region. I have taken the one before it. You can take around it. It depends. And then this we again put it on reverse complement. Then we get the reverse complement of it, and we again map it to ensemble, where we can see this is the position where the sequence lies. This would be the position where the change is happening, and this would, the complement of it would be the actual nucleotide. So C, it shows here C, but actually it is a G, and that has changed to A. And if we count, it would be this 113 would come here if we count from here. So this is this is actually 209 113 384, and if we count from the this direction, it would be 3113. And the cDNA level, we have C, the CGT, where C is getting converted to T, and therefore arginine at this arginine 132. So you can see this is 133, so this is 132, where it is getting converted to, because it's getting a TGT, which is a cysteine. So this is the result that you'll get in this case. Moving on, another case of acute leukemia, where bone marrow morphology and flow cytometry was consistent with T-cell acute lymphoblastic leukemia. NGS just showed a Notch1 mutation. So Notch1 mutations are very commonly seen in TLL, and this paper by this study by Brady showed that TLL can be seen up to 70% cases of Notch1 mutations can be seen there in TLL. And recently, in 2023, we had this NGS-based classifier risk stratification of TLL based on Notch1, and they said that low-risk classifier, if just Notch1 mutations are there, so it would be a low-risk classifier in the absence of NRAS, DNMT3A, IDH12, IKZF, TP53, and PIK3 pathway mutations. So this case would also be a low-risk TLL. So how do we do the nomenclature for this Notch1? If you have entered, then this local anti-hero would, how many of you got this? Yeah. So I'll show you manually again. There is a change A to T, and we take the sequence, put it on reverse complement, and then we check it on our ensemble ID, and their ensemble website, and there we see that this is the position where the change is happening. This would be this 9410 position, and here the A is getting converted to T. The complement of it is showing here, so this is actually A here. The rest of it would be just as we've seen earlier. GTG is getting converted to GAG, and the valine is getting converted to glutamine. So this is the final result that you're getting in this case. Okay, that's it. I'll now call upon Gojiri to continue with the session.
So till now we saw point mutations. Gojiri will be covering the in-frame and frame shift changes that are there.
Yes. Good afternoon, everyone. I am Dr. Gojiri, and I am here to complicate matters further for you. So let's start with this case. This is a 62-year-old female with anemia. Cytogenetics showed deletion 5q, and on NGS we got mutations in DNMT3A, TET2, and PTPN11 genes. So amongst this, we'll look at how to do the nomenclature for this TET2 gene. So we see here TET2, which is located on chromosome 4, is showing T at this position 106157841, but the altered sequence is now showing TG. Now let's look at the integrated genomic viewer for this, and we can see that this is the position 841 which was seen in the reference sequence. This is the next position 842, and we can see that there is an insertion G in here, which is a frame shift insertion, right? So when we map this to ensemble, again we see that this is the genomic coordinate here, which corresponds to 106157841 to 7842 insertion G. Now my question to you all is, which one of these options is correct when we do HGVS nomenclature? Is it option 1 with insertion G, or is it a duplication? I'll show you the IGV again. Okay, so for frame shift, basically when the insertion or the inserted or the deleted sequence is divisible by 3, we'll call it an in-frame or non-frame shift. Okay, so basically the reading frame of the coding DNA will not shift, because 3 base pairs code for 1 amino acid. The reading frame will not shift. That is why it will be an in-frame or a non-frame shift insertion or deletion. When it is not divisible by 3, then the reading frame of the coding DNA will change, will shift. Hence, all the amino acids that it is coding for will also shift. We'll see the examples further. So basically we look for the genomic coordinates that we are getting on the NGS results, right? So at this particular genomic coordinate, we see that there is an insertion here on our sequenced data.
Just one base pair getting inserted. So that is not divisible by 3. So that is frame shift. Automatically it will lead to frame shift.
I will show you in the protein coordinate how the frame is shifting. Okay, so coming to the question.
It should be insertion G.
Any? So sure? Confident? सही जवाब. So let us look at why duplication is the right nomenclature. So a duplication is defined as a tandem copy of an upstream sequence. So in this case, a G was present already upstream of where the insertion has happened. So it is actually a duplication. The position of a duplication is represented by defining the position of the nucleotide that is duplicated. And for variants in stretches of repeated DNA sequence, the 3' most rule applies here. So the 3' most position possible is arbitrarily deemed to have been changed. We'll come back to this. So we can see here there is a G here, right, at 4,2. So it is actually the G at 4,2 position which has duplicated, right? And when we come to further DNA level descriptions in HGVS, we see that for deletions and duplications, there is it's optional to specify the deleted or duplicated nucleotide sequence. However, when we come to insertion, the specification of the inserted nucleotides is mandatory. Okay, so now coming to the cDNA coordinates or the transcript coordinates. At cDNA level in ensemble, we can see that this is the position where G has gotten inserted or rather duplicated at position 2743. Okay, and now let's come to protein coordinates. So here we can see at position 915. So this is the position 914 where CAA is the coding DNA, and after that, sorry, this is the position 914 where CTT is the coding DNA for leucine, which remains same. After that, between G and GCT, there is an insertion of G, right? So we can see that alanine, which was present in the original sequence, has been substituted by glycine. And now we can see that because of this one insertion of G, the entire reading frame has changed. All the amino acids have shifted. And on top of that, there is a termination codon at the 9th position from where the change has occurred. So if we can count, this is the change 1, 2, 3, 4, 5, 6, 7, 8, 9. So at the 9th amino acid, there is a termination codon which has come up. So there is a frame shift termination at the 9th position. So like Shweta ma'am has already explained, we prefer the three letter nomenclature. So alanine at 915 substituted by glycine, followed by a frame shift termination at the 9th position. So if we put this in, we can obviously, it's easier for us to put it in variant validator and get the answers, but this is how manually we get to the answer. If you're using the if you're putting it from the Excel, please go on the Kava sheet. No problem.
One more thing I would like to highlight here is that you can see that genomic variant is a duplication, the protein variant is a frame shift, and the transcript is a duplication. Right? So this is one more important feature of HGVS is that they demand that you should be independent at each level. So when you're looking at protein, you do not need to look back at the DNA what's going on. So you should just look at what's happening in the protein level and say it like that. So that's also another important thing. What I'm saying is HGVS is independent at each level. So for example, here duplication is happening in genomic as well as in transcript level, but in protein level it's a frame shift, right? It's a substitution basically which resulted in frame shift. But the fact is that you are supposed to say like that only. So at each level you should be independent of upstream or downstream level and only look at that level and do the nomenclature. So that's also another fact. Correct. Duplication, yes.
Okay, so can we move on to the next case? Yeah. Okay, so this is the next case. This is a 64-year-old female with complaints of loss of appetite and weight since 1 year. There is presence of splenomegaly on examination. Peripheral blood smear showed leukoerythroblastic picture. Bone marrow aspirate was dilute. And like we would expect in most cases, bone marrow biopsy showed a grade 3 fibrosis on reticulin stain. Now, when we did NGS, we found a 5 base pair insertion in the CALR gene, which is a type 2 CALR mutation or variant that we see in most of the myeloproliferative neoplasms. And now, this 5 base pair insertion, let's look at the integrated genomic viewer. So, this is the nucleotide position 13054 and 628. And there is an insertion of 5 base pairs between these two positions. Okay, so similarly we can see that at the start and end there is a 5 base pair insertion between A and G. Okay, again a frame shift. Yes.
So when you're putting your input into the variant validator, for substitutions it is easy, like
Yes.
ek se ek ho gaya.
Yes, sir.
But for when there is a long change, so that AG is converted, so how, and the coordinates are also not the same, start
Yes.
and end is different.
Yes.
So how do you put your input there? I mean, that is also, so you put a hyphen or you put a underscore, how do you go about it?
Yes.
Question.
Chromosome number, we put start
codon.
codon, we put a hyphen, then the reference. So suppose reference is nothing, something
Can you show
insertion
one
has
of them
happened.
for all of us, one
Yeah.
such example, so that, I mean, like,
Well, let it show. Google is working.
So go to that NPM one.
Again,
wala
that thing will
sheet
change
share
side
or
to side
downloads,
also.
yeah.
So every side, if you go there, they will give a directive how they want their input to be.
Okay.
So, for
Anyway,
example, there are some sites which
and
will show
another
tomorrow.
question is that if I have an Excel, so can I put all it together and then it gives out a list again?
No, so variant validator has a format by which you can okay.
Batch
So
validator.
Batch validator is there, but you will have to give it in that specific format, like start codon, then you can do that. Batch validator can be done
मतलब
together.
एक बार enter करने के बजाय if you have Excel
You can
and
do
support
that.
it
You
and
can
then-
do that. You can do that.
Kawa
So
wala
ये NPM 1 वाला ये TCTG वाला 13
अच्छा, okay.
जो है हाँ 13th हाँ इससे जो नीचे वाला है क्रोमोसोम
यही वाला दिखा दो कावा वाला ओके
How to put it in? उतना ही दिखाओ खाली. How to put? There is nothing in reference sequence, no. So how to put that?
So this is a type A insertion of NPM 1. So she's opening variant validator and she'll be putting that online. Try it out. So basically variant description. So after chromosome 5 there will be a hyphen and there is no reference sequence. So there will be 2 dashes in between the chromosome start, and because there is no reference sequence it will be dash or hyphen followed by a hyphen again and then TCTG insertion.
Show the full thing.
So GRCS and then submit. So if you look at the way we can enter the variants, there are multiple methods. Here enter the variant description as follows. So they have given you can enter in any format that you want, and then if you select this you will get what you are getting. This is the way it is done for insertion. For
the
can
dash
you
will
show
be-
one for deletion? So yes. So it is just that in the reference sequence nothing was there, so that is represented by hyphen. For deletion, it can be a single hyphen also because we are not giving any nucleotide after that.
Yeah, we have given a Kawa
yes.
sheet.
We will when we come
We'll
to
talk about
NPM 1 example, we will be discussing about that. For deletion, you can have a single hyphen after this thing. You can put either single or double hyphen, it doesn't matter, you will get the result.
Just go to the home page एक बार whenever it is. या नीचे? नहीं sorry, try it out में आता है ना, try it out then नीचे आता है. I think. So they will give you all this as well, no? So they will give you all this format in any of the format you can put. So they will guide you. It's not that.
So it
you have to
can be
know.
hyphen, it can be colon.
So all
also,
these formats you can put it like.
it is acceptable.
Yes. And this
yes.
may be slightly different side to side. So in that side you have to see what kind of format they take.
Okay, so moving on. So we had got this particular 5 base pair insertion. Okay, we map this on ensemble and we get the genomic coordinates, which is 130546227. And as we had discussed, the range should be given when there is more than one nucleotide insertion or deletion. So 54627 to 54628, there is an insertion of TTGTC. Okay. Next, coming to the C coordinates. So again, similarly, we can map this particular sequence here, which corresponds to 1154 to 1155, and it has to be followed by insertion and specification of the nucleotides which have been inserted. Now coming to the protein coordinates. So we can see this, the upper one is the original sequence or the reference sequence, and the lower table is the altered sequence. So what we can see here is that after this AA, there is an insertion of TTGTC, which has led to a frame shift of the coding DNA or the reading frame, and hence what has happened is in the reference sequence there was these amino acids, and at 417 position there was a stop codon in the reference sequence. But because of the shift of the reading frame, what has now happened is at 417 position the reading frame has shifted, the stop codon is changed to AGG, and now the stop codon has shifted to the position 432. So there is a frame shift termination after 47 amino acids after the change that we see. So if we put this in HGVS, we can see that this is the results that we get in this particular insertion. Coming to an in-frame insertion. So this is a case of AML with a SEPA mutation or a in-frame insertion in the BZIP domain, which is a very common SEPA variant that we get. And let's again take a look at the integrated genomic viewer. At this particular position 2368, and between 2368 and 2369 there is an insertion of TCA, which is an in-frame insertion, and we will see why it is in-frame and how it affects the reading frame. But before that, this SEPA gene is located on the reverse strand. So we also have to consider which strand the gene is located on before we look at the nomenclature. So like Shadma has explained already, we put it in the reverse complement tool, we get the reverse complemented sequence, and we map it on ensemble, we get it somewhere here. Okay, so it's actually not left aligned and right aligned, it would be forward and reverse complemented sequence. So we this is the sequence that we saw on IGV. So between G and the next T, there was an insertion of TCA, right? Followed by another TCA which was originally present in the reference sequence. So this is not just an insertion, what is it? It is a duplication of this particular sequence. But because it is present on the reverse strand, we'll have to swap it, right? So our TCA which is inserted here gets converted to AGT. We are reading it on the reverse strand, so it is converted to AGT, which is a duplication of the next AGT which is found here. Correct? So the genomic coordinates are this, from 69 to 369 to 371, there is a duplication. If we look at the yes.
After reverse thing, it is not.
No, so if you see AGT is again duplicated.
Excellent question. Yes. Actually, so what you are asking is maybe there are 3 stretch of repeats. Okay, so one of them is duplicated. So the number 4 you will now point according to 5' to 3' rule. है ना? The number 4 is inserted, so the number 3 has been duplicated, right? So 1, 2, 3 like that. But in the reverse strand, it is now changed. The direction has changed, right? So now the number 3 here will again be duplicated, but it is actually corresponding to the number 1 in the original strand. I think that's what you were asking, right? It has changed. Yeah. So let's say this is the 5' strand, okay? So yeah. After reversing. Again, independent of the primary thing.
Yes.
So when we are talking about the coding DNA, you are independent of the genomic. So for example, here, if this should be taken, right? So let's say this is the actual genomic DNA, okay? So I will say that this TCA is the one which is the most 3', so this is what I will take. Maybe this is corresponding to this, and this is corresponding to this. But here, when you are reversing the thing, so I will say that this is which is the most 3' one which should be selected. So that way. So at it is possible that the number 5 is actually here number 7, but that is what it will happen, because we have to choose the 5' to 3' rule at each level independent of upstream or downstream.
So at genomic level also it will be separate nomenclature will come, and at coding level a separate nomenclature will come independent of each other. We have to apply the 3' rule independent of it is in reverse strand or forward strand.
It may
It
seem
may
very
be different.
counterintuitive, very stupid, but this is how it is. Correct.
Yes.
Yes. Yes.
Yes, 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 yes.
ह्यूमन कर दोगे तो ये ह्यूमन.
Yes.
In which direction we will get the sequence and why we need to reverse? Because mostly we will be getting in the forward direction.
Benevolent, you will anyway get it, so that is true. You can get it from that way. But if you have to look at the genomic coordinate, then you will have to reverse it. Correct. Then only. Otherwise, no. Chalo, let's swap.
Okay.
Ensemble में होता है reverse strand का information. Yes. Yes, yes. And if you try to map it without reversing for reverse strand, it will not map. Then you will have to think that why this has happened. Then you go and see it is reverse strand. It usually happens that. Ha. Yes. Yes.
Okay, coming to the
I'm getting a different answer in variant viewer for this case.
Because we had used Kava for ये. हाँ, Kava. So Kava नहीं हुआ. It will come, है ना. We will come to that with her case, Poorva's case. So just wait for some time. Once we explain, that will come.
Okay, now coming to the cDNA coordinates. Here is the insertion of TGA or duplication of TGA, and it maps to, if we count here, it maps to 952 positions which have been duplicated, again applying the 3' rule. This is the cDNA coordinates that we get. And now if we look at the protein level nomenclature, here we see that at 3-1, this is again the upper sequence is the reference sequence, the below one is the altered sequence. We see that there is an insertion TGA at this position in cDNA coordinate. So now what we can see is at 316 up to 317 amino acid position, the amino acids are same. After that, because of insertion of an extra TGA, it has led to an insertion of methionine. But if we look at the next sequence, that is amino acid TSD, we can see that again the reading frame is preserved, the next amino acids are same, right? So what has happened is there is an insertion of methionine between position 317 and 318 because of a 3 base pair insertion, right? So how do we do the nomenclature is leucine at 317 and threonine at 318. In this range there is an insertion of methionine. Okay, so if we
with respect to the one what you said, like if it's divisible by 3,
yes,
Yes.
then basically now even if it is divisible by 3, in this case of course the subsequent codon was translating
Yes.
to the same protein, so it's
yes,
fine.
yes,
If it is not translating to the same protein, here it's like after that M methionine, it's TS, so it's matching with that. Only there is an insertion of methionine. Like
yes,
if the next codon has changed, instead of ACC, it's something else, and it's not coding for T protein, then still you call it as. So my point is very simple. If it's divisible by 3, can I just say
Yes.
yes,
in
yes.
though if the proteins after that are changing?
Usually that
You-
doesn't
it
happen.
will not happen.
Yeah.
There is another in-frame deletion example where we can also see that
Okay.
the reading frame gets
But you
preserved.
got my question right?
Yes,
Yes.
I got.
Okay.
Yes. So, but usually that doesn't happen because after 3 nucleotide, usually it will either change 1 or 2 nucleotides and others will remain same. Usually that happens. Because, within frame, that usually happens. Because, see, 3 nucleotide are inserting or deleting. So, it will affect the 2 consecutive amino acids and not affect beyond that. Because it is change in only 3 nucleotides. So usually these 3 nucleotide change will not. Sorry?
Because why I'm asking is like here it's very clear now. ACC, AGT has come, so only the methionine is inserting. Now if it's not ACC and if it's coding for some other protein which is not T.
So that is what. So basically maximum what will happen is 2 amino acids will change. Like M and T will change. Not apart from that. Beyond that it won't change. Because it is just 3 nucleotides, right? So it will maximum affect 2 consecutive amino acids and the reading frame won't change. That is why we have to call it in frame only. Yes. Yes. तो वो अभी इससे previous जो example दिखाया था, go back to your वो. So it becomes, see, here what happened, the frame has changed. So what is happening is your amino acids are changing. This is the reference, like how it is supposed to be in an original sequence. So here these insertion are happening. All the amino acids are changing. What was supposed to be stop codon at this 417 position of nucleotide of amino acid, now at 417 there is arginine that is happening. But what is the next stop codon that is coming? Okay, from this change, like from N 385, the next stop codon is coming at 432. So for naming this, it would be lysine that was supposed to be there at 385 position is now changed to aspartic acid, right? Aspargine, and frame shift. This whole frame is shifted and termination is happening at 47th amino acid of this. Okay. Frame shifts.
Yes.
This is how we will describe.
From the change.
From the change.
From here, this will be the first amino acid
1.
that has changed. So this will be 1, and then the 47th amino acid that.
Yes.
So it's like, how will it affect the function then? I mean, like what gene
So, but then it won't produce functionally effective gene, right? The function that it was supposed to do, these all amino acid may not do the same function as it was originally supposed to do. Even though it is longer, it is not meant for the purpose. Yes. And then we do always see in silico prediction, pathogenicity, is it causing any change or not? That and all is a separate thing altogether.
Okay, moving on to some examples of deletion. Now let's see how deletions are annotated. So this is a case where there is a U2AF2 deletion. We'll just we mapped it on ensemble. We found the genomic coordinates here. We'll see how we look at integrated genomic viewer. Okay, so this position is 56173,949. So this is 950. From this position to the position here, that is 964, there is a deletion of 15 base pairs, right? So it is an in-frame deletion, right? Now these coordinates that we found, 56173950 to 3964 deletion, we'll put on variant validator and we'll see what the answer is.
Some of your variants are like homozygous. I don't know whether it is right to call them homozygous or like the variant elite frequency is close to 100 or something.
No, it is just a
sorted the base by change. So she's only showing you the uppermost bases which are changed. So it's not like.
So there will be the rest of them.
One could be, yeah. Well, maybe in that case it was like that. But here also we must have sequenced many more reads than this. So she's just showing you the sorted bases. Usually in our work we get heterozygous only because we are anyway detecting somatic changes in cancer. So we very rarely get homozygous changes.
Okay, so our genomic coordinates that we found on integrated genomic viewer are this, but on variant validator we are finding some other coordinates. I'm sure all our halat is like this right now, right? Let's try to figure out why. So what does the 3' rule say? The 3' rule says that the 3' most alteration which results in the final altered sequence is arbitrarily deemed to have been changed. So when we see at the original alteration that we are seeing on integrated genomic viewer, this is the deletion which has led to the final sequence of CAGAATT at these positions, right? But if we look at this entire sequence, there is another sequence which is more 3' to the original deleted sequence, which is giving us the same final result, right? If we see that if this particular segment is deleted, it is also leading to CAGAATT, which is the same as the final sequence in the upper example that we see. Hence, by convention, by 3' rule, we say that this particular 3' most segment is the deleted sequence and not the one that is being shown in IGV. So the coordinates now shift to 3'.
So this is the problem is between NGS, VCF and HGVS guys. So they do not agree to come to this common ground. I don't know why. This has been discussed many times. IGV, so IGV जो दिखा रहा है वो NGS VCF से जो आ रहा है वही वो दिखा रहा है. वो भी ऐसा नहीं है कि it is like something God is telling or something, no? That's a problem.
That is what. No, so we want to make you aware that it is not what comes in IGV.
Main problem is that why this NGS, VCF and HGVS guys are not coming into same term.
Yes.
The thing is that HGVS was a long back organization. They made this 3' rule, and NGS has now started this 5' thing. So they HGVS are adamant that we won't change. Why would we change for you? So that's the main problem. So we are kind of stuck between the two.
All the annotation, usually all the variant callers, they will follow this 5' rule for NGS instead of 3' rule. So what you will see in IGV will be what is that 5' has been followed. But when you manually derive it, or if you put in variant validator, there are few tools that are there. Like Kava is one of such tools that gives you a 3' aligned sequence. So when you know that you are dealing with such deletions, insertions, and you have to give 2-3 things are there, you can go to database and see, like, if NPM, for example, for NPM1 type A, if it comes, you will get a wrong sequence when you look at the variant callers. Like somatic seq will give you a wrong annotation, but if you look at Kava, it will give you a correct. So Kava is it follows 3' rules. So that way you should be aware what can be possibilities and how to go about it. You can go to Cosmic database and check how they have given nomenclature and see if what you are giving is correct or not. That way.
Yes.
Yes, so NPM1 case will be coming, and here what is happening is, instead of we predicting it, like here this is the resultant rule that is there, resultant sequence, like we are getting CAGAATT. Okay, this can happen because of deletion of either the sequence above or below. Yes, anything will. So when we have the same consequence, we will take the one which is following the 3' rule, which is right aligned. Okay, where the sequence change, yes. When we are getting the same result, we have to follow the sequence that is getting deleted at 3' end and not at 5' end. C level also, and independently, yes.
Whenever we face this challenge, we have to follow.
Yes.
In this case, when we are searching in ensemble, I'm not getting that main or canonical.
So what I'll do is, we had just put in the Excel where we had put NMIDs that we are using, so that it will become easier for you people, because there is some issue with ensemble. This is not the frequency that we don't get this thing.
You could go to MenSelect website also.
Yes.
Which is the
yes.
transcript for this particular gene.
Because these are common genes only. For these also it is not giving. There is some issue with ensemble today. Usually this doesn't happen. Ensemble has selected this day.
We have favored ensemble too much today, I think. Like UCSC guys will be surely cursing us. Why are we using ensemble? Why not UCSC?
So, नहीं. So basically, what input you are giving to variant validator, it will give you output according to that. So input, if you are giving from somatic seq, that is your following your 5' wala rule, then it will give you incorrect output. So you should be aware of that. Frame Kava and C. ऐसा सिचुएशन, इंडेल्स, डिलीशन, इंसर्शन, ये सब के लिए कावा पे देखना है आइडियली. डिलीशंस, इंसर्शंस, इंडेल, कॉम्प्लेक्स इंडेल, वेरेवर. हाँ हाँ.
सिचुएशन में देयर इज नो प्रॉब्लम ऑफ 3 प्राइमिंग, नो, एक ही डीएनए जा रहा है, एक ही न्यूक्लियोटाइड जा रहा है. सो
I have one doubt. So when I'm using this variant validator, it is giving some warnings, like some with the VCF specification of 4.0 and then 4.1.
Gojiri, I think you can continue.
Okay.
Otherwise, I don't think.
Okay, so we'll just quickly go through the cDNA coordinates. We saw what was the sequence that is deleted. We mapped it on ensemble to this particular position, 572 to 586 deletion. No more twists here, right? And now we'll look at the protein coordinates. Like, sir, how you are asking whether in-frame will always lead to the same frame. We'll see another example here. So this was an in-frame deletion, right? So we see that this is the, this is the sequence that is deleted, correct? So what we can see here now, this is the reference sequence, this is the altered sequence. From here to here, there is a deletion. So from I, there is deletion. Because 15 base pairs are deleted, 5 amino acids will be deleted. So I, N, D, Q, D, K are deleted. And now the next codon which is forming is AAT rather than ATT. But if you look at the codon, the AAT is still coding for N, and the further frame is still preserved, and the reading frame is preserved. Right, so there is a deletion between 191 and 195 position on protein. And when we look at variant validator, we are getting the same results. And I'll be discussing this last case by me. This is an frame shift deletion where a single nucleotide C is deleted in TET2 gene. We'll quickly go through it. We can see that from ACC, there is a deletion of C, and it is leading to this particular altered sequence at 575 position. But again, now we are following the 3' rule. So this C is not considered deleted by HGVS. In fact, this is the C that is considered deleted, and hence our genomic coordinates shift by 1 base pair. So 6576 deletion. This is where we map on ensemble. cDNA coordinates, same thing. We get the coordinates, and on protein coordinates, we see that there is a shift of the reading frame because of 1C deletion, and then there is finally a termination at 58th amino acid after the position. We map it to variant validator. Did I say it's the last case? Yes. No. I invite Dr. Poorva to further discuss the next cases.
So I'll continue the discussion further. So this is the peripheral blood morphology picture in a newly diagnosed case of AML. What peculiar feature can you appreciate here? Yes, so most common defining genetic abnormality associated with it is NPM1. So it is important for us to know the nomenclature of NPM1. So we'll go to case 13, NPM1 type A frame shift insertion. So this is what somatic sec Excel file is giving us. The genomic coordinates, reference sequence, altered sequence, the gene name and cosmic ID. So when I put this on variant validator, so after selecting the NMID, I'm getting this nomenclature, 542 and 543 insertion TCTG, and the transcript variant I'm getting is 858, 859 insertion TCTG. Let's go ahead.
Protein is at 287.
Yes.
Important है क्योंकि ये mistake हम कर चुके हैं.
Yeah. Protein variant leucine 287. So let's go ahead. The integrated genome viewer is showing insertion TCTG between C and T. So our reference sequence will be TC Then TC, TCTG, TCTGG. So we go to Ensemble Genome Browser, search the IGV sequence. It is getting mapped here.
We already have an
So-
answer. Correct answer. बोल दिया.
हाँ वही दिखा रहे. So the genomic coordinate of CT is 543, 544, and TCTG is getting inserted between C and T. So the genomic variant of this insertion will be 43, 44 insertion TCTG. Then same with transcript variant identification. The transcript of CT will be 859, 860. TCTG is getting inserted between that, and transcript variant of this insertion will be 859, 60 insertion TCTG. In protein variant identification, this is the original sequence, and TCTG is getting inserted between C and T. This is the protein number 287 of leucine. So the frame is getting shifted from leucine, and the altered sequence will be C, TCTG, T, and we are getting TAG stop codon on 13th amino acid. So the protein variant of this insertion will be leucine 287 frame shift termination 13. So this is what we have manually derived. So let's check on Cosmic database as well as variant validator. So Cosmic database is basically catalog of somatic mutations in cancer. It's a comprehensive database in which we can see various pathogenic mutations, and we have IDs for each variant. So we put Cosmic ID here from the Excel file. So as we can see here, the nomenclature we are getting in variant validator differs from what we are getting on Cosmic ID. As well as same, the transcript nomenclature we are getting from variant validator differs from what we are getting on
Just because, just to clear this confusion, because people will think like that only, na?
So basically there was mismatch between the Cosmic database and our variant validator that we were getting. Cosmic database is something that has been given nomenclature properly, and we know that many people have reported. So why was this?
So what could have gone wrong? So let's go back to IGV again, and here we can see that the insertion is between 43 and 44 TCTG, and we have seen the altered sequence here. So the first mistake we did was we didn't follow the 3' rule. So 3' rule states that for variants in status of repeated DNA sequences, the most 3' position possible is deemed to have been changed. And the second we didn't follow was variant description. As Sir said, it was possible according to two classes, a duplication and insertion, but the priority assigned is for duplication. So according to HGVS nomenclature, this will be the original sequence and this will be the duplicated sequence at 3' end. So somatic seq file is showing us this altered sequence and this nomenclature. However, in these cases of deletions and insertion, we need to check on other variant annotation tools like Kava, which are right aligned, that is they follow 3' rule. And here we can get the reference sequence and altered sequence as well as the correct nomenclature.
So if we put the input of Kava sheet, so when I have circulated the Excel sheet, I have circulated two sheets in the same Excel. The second sheet is the output from Kava. If we give that input to variant validator, we will get the correct nomenclature. So basically, we have to be aware that we cannot blindly accept what NGS report is giving, and like when we are in a hurry, we just put the coordinates, we put in variant validator, get the results, and we think that we are done with it. But we have failed EQUAS because of this reason, that we had given type A insertion according to the 287 code that we were getting in the output of somatic seq. Then we started taking care of that we are putting all the insertion indels, we take output from Kava, and then we get this results, and we incorporated Kava also after that only. So Kava is another sheet that we get, after our bioinformatics pipeline. So this is how it comes in Kava. It is not a separate, so it is a bioinformatics tool. Like Somatic Seek is a variant caller, Kava is also another variant caller. In our Excels, we get an output of Kava because we have incorporated that in our bioinformatics pipeline. Because for us to manually sit and do for every case, it is not possible at all. We have to keep reporting many cases. It is not possible for us to keep checking. So now we know that Kava is a tool that follows this 3' rule. So for all the indels, we go back to Kava and take the input from Kava instead of taking it from our Somatic Seek output. That is our the sheet where most of the point mutation and all we take output from Somatic Seek itself. But for specifically for indels where we are dealing with insertion deletion, we take output of Kava.
So whenever you are doing your color also just indel.
Yes.
It's always cross check with Kava and give it.
Yes. Yes. Yes.
Either do it manually or look at Kava.
Incorporated in our pipeline. So we have incorporated this in our pipeline so that when we get the Excel.
Yes.
Yes.
Yes, but it's not like an online tool where you can do it. You have to download it and get it integrated into your bioinformatic algorithm only. So it's not like something where you can just put the sequence and it will give you.
Huh.
For our hands-on users, we cannot implement with the pipeline. Okay. We get only that output files.
There will be a way, I'm sure. So you just have to go to that Kava web, their main papers. It must be in GitHub or something, I think, Kava, no? So you should go there and check. There must be a way out, I'm sure. Ideally, yes. Any indels. Any indels.
Deletion, duplication, anything, you should be doing this.
So when we do it manually, we are getting the same variant as shown in variant validator now. So let's go to type B, frame shift insertion. So this is what our Somatic Seek Excel file is giving us again, the reference and altered sequence. So when I put it on variant validator, it is giving me this nomenclature. But now we know that we have to check on IGV. So this is the altered sequence TC, TGCA, TGG. And we know that we have to follow Kava for right alignment. So TGCA is getting inserted between C and T. According to HGVS nomenclature 3' rule, this will be the original sequence and this will be the inserted sequence now. So manually also if we derive,
I think we can skip the manual part.
Okay, fine.
Yeah, for the interest of time. So basically, again, we always look at these variants in the in Cosmic database, what it is being reported and what is coming as output, and we try to match it wherever there is discrepancy and see.
Now everyone will say, "Ha ha, samajh gaya." Ha ha. So now I request Dr. Narasimha.
But
type D and type I, but similar to type
A and B, the two most common.
Commonest ones.
So I request Dr. Narasimha to enlighten us with his session.
Thank you, Dr. Poorva. I know I'm standing between you and lunch, so I won't take much time. So I'll just take 5 minutes. For the first case, I just want you to concentrate directly. No need to use your laptop. But for the second case, I want you to use the variant validator. So mine is very simple. FLIT3 ITD. I'm going to talk about FLIT3 ITD. So ITD stands for internal tandem duplication. So internal means it is occurring within the exon. Tandem means it's just occurring next to it. And duplication, we all know it's the same sequence. So yesterday we read about KMT2A partial tandem duplication, which means it occurs across multiple exons. Whereas in FLIT3 ITD, it is occurring across only one exon. So here, this is the output which we got. Now, when we see the first we'll see what is duplication. So this is the sequence. According to HGVS, when we say duplication, the duplication occurs to the 3' end. So when we have to label this, we will write the coordinates from this position to this position, and duplication means it occurs to the 3' end of it. So this is the IGV file we got, and this is the sequence. This is a 75 base pair insertion here. Then when we see in ensemble in FLT3 is in reverse strand. So the position comes to here when we map it to the in the ensemble. But we know that FLT3 is in the reverse strand, so we need to reverse complement it. As we know that we have to go to this website, we use the sequence here and then we get the output here. Now we see where it is getting mapped. But one warning or something which we have to keep in mind is this in ensemble, this corresponds to 60 base pair length. So whenever we are using FLT3 ITDs, whenever we get some FLT3 ITDs, if it is more than 60 base pair in length, when we copy paste the entire sequence, we may not get the exact match. So what we have to do, we have to start deleting from one end, either from the top end or the bottom end, so that we will get the 60 base pair length. So for example, now I am starting to delete from this side. I got a 60 base pair match. So I know that there is a few base pairs above and few base pairs below, and now I know that from where to where the codons are duplicated. So when we see it in the genomic coordinate, it is from 23 to 97. This number corresponds to 97 because it is in the reverse strand, and this corresponds to 23. So this from 23 to 97 has been duplicated. So the same with cDNA. So from here, this position is 1759, and then this position is 1833. So from this position, this position has been duplicated, and the same for the protein. So from 587 to 611. So when we use manual method or variant validator, we get the same result with this. Okay. So when you can also confirm it here, it is showing duplication instead of insertion, because duplication will get a higher priority over insertion. So this is the final report which we get. We have the refseq and we have the coordinates. So it looks like it is very simple. FLT3 ITDs is very simple. But the second case which we are going to discuss now is like a Christopher Nolan movie. I just want you to copy the case 18 in variant validator and check what results you are getting. Case 18. But it's a good learning experience, because in this one case, we will learn almost all the HGVS rules. So if we are able to understand this case, we will understand most of the HGVS rules today.
Wow. Very loud claim, no?
So when we see the IGV, again, we see where the insertion is taking place. So the insertion is of 33 base pair. The sequence has been given by the IGV. Now again we map it with the HGVS nomenclature, with the ensemble, and then we see the insertion site is happening here. We are reverse complementing the sequence. We got the reverse complement. But when we put it, when we check it with the mapping again, so almost all the sequence gets matched with it except for this A, which is getting circled here. So when we put it in here, the ref is A, but the sequence in our the sequence which we have is T. So the T is being replaced, the A is being replaced by T here. So that is why when you put it in the variant validator, you will not get it as a duplicate. Instead, you get it as an insertion. So this is something which we came across during the preparation of this workshop. So
because duplication has to exactly match.
Exactly match. Sequence. So that is one point here. So when we talk about duplication, the entire sequence must match. But here, the last nucleotide is totally different. That results in we have to put it as insertion. That is one rule of HGVS which we can apply here. The second rule here is, so when we see the insertion here, TGA, TAA. So when you see the protein pamphlet which we have given to you, can you please see what TGA and TAA codes for? Stop codon, right? So when we see this, then how to explain this? So this TGA, TAA both codes for stop codons. But in FLT3 ITDs, we know that it's not a stop codon. There is no stoppage of transcription happening here, right? So basically what happens is that here, when you see here, this A is the last nucleotide of the previous protein. And this GA is first two nucleotides of the last protein. So when this duplication... No, this G and A nucleotide protein
amino acid.
Amino acid. So this GA are the first two nucleotides of this amino acid, and A is the last nucleotide of the first amino acid. So when there is a duplication, these two will go and join with this. These two of the original sequence will go and join with the T. So when you see the sequence, even though here it tells TGA is for stop codon, this GA should also be taken into consideration. So the exact insertion of proteins is GAT. So that codes for aspartic acid, and then GAT again, that codes for aspartic acid, and then the sequence is the same. So when we, that is why I think in our report we should include all the three genomic level coordinates as well as cDNA coordinates.
कोई वो नहीं है.
So that is why in our report it should have both genomic DNA, coding DNA as well as protein, because if you give the coding DNA alone, the TGA and TAA codes for stop protein. So that is why we wanted to confirm here. And then one more thing I wanted to tell here is, so the question given to us was at 68 position, but whereas when you put it in the variant validator, it comes to 300 to 301. Can anyone explain why? This is again the 3' rule. So the same sequence is there in the reference, and this almost the same sequence gets inserted except for the last nucleotide here. So the 3' most position will have to be changed. So even though the insertion happens at 268, the variant validator will apply the 3' rule and give you the final answer as this one. So
corresponding to the protein, new protein made
Yes.
Yes. So
But I think the real question will come from Anand sir probably, that is it an ITD or not? You are talking it as an insertion, no?
Insertion,
the last
yes.
one was duplication.
Yes.
Should I give क्या बोलते हैं उसको midostaurin or not?
So technically speaking, it is like a duplication only, but as per the nomenclature, it is not exactly fitting with the criteria.
Terms are legacy terms like ITD. This is a very legacy term, but this is for all practical purpose definitely ITD. Although according to HGVS, this will be an insertion. So, for example, we called biallelic SEPA, but none of us has actually proved it that they are in two different, we have not phased it. It's basically double mutant, but we call it biallelic. So, these are legacy terms. So, we'll call it ITD only, but according to HGVS, it is insertion.
Yes. So, that is, this will be the ITD.
ITD, yes.
So, thank you.
So, now I invite Dr. Saurabh to talk about complex indels.
Motivated by Christopher Nolan, it seems, you guys.
Thank you, ma'am. As for Dr. Narasimha also, I am quite aware that I am also standing between you and lunch. Like these beautiful cases are lying between the beautiful memes that we have put. So, and there were many queries regarding to Delins. So I want to clarify one thing, that as I speak about Delins, the HGVS is currently updating its nomenclature regarding Delins. So what I tell you here might change till the end of the year. They have added some recommendations. We will see what are these. So the old term for Delins, like Neha ma'am also mentioned, it was indel, but it was quite a confusing term. So because we are not sure whether it is an insertion or deletion, insertion deletion simultaneous like that. So we used to use it in multiple purposes or in multiple manners. So HGVS said no, it is a very confusing term, it should not be used, and they gave us a term called as Delins, where a sequence change, where compared to a reference sequence, there are one or more nucleotides are replaced by one or more other nucleotides, and which is not a substitution or inversion. So coming to my first case, a 70-year-old male, fever with mediastinal mass, classical history, flow cytometry reveals it's a TALL. Now let's see what we got on NGS. So in the Excel file also you will see we have got these two mutations. So the question arises that whether you should treat these PTEN gene mutations as a single variant or two different variants. We can see that their coordinates are very much closer. Their VAFs are also very much similar. And PTEN gene is a part of PI3K pathway, which is now has been established as a prognostic.
Excellent question. He is asking for IGV plots.
Yes. Next slide is IGV.
That will come. Yes, that will come ahead, sir.
Because whenever the complex variant occurs, it shows as two variants, especially if the coordinates are very
close. Yes, sir.
So that is why we showed this case for that purpose only. Next will be IGV only.
So when I see this on IGV plot, I see that at this position 897171, there is an insertion of 4 Gs, and just next to that, this C is converted to A.
Now, how would I say that if I had insertion in this and A here, then I would say it is two separate mutation. But what we are seeing here is everywhere where we are getting insertion, in that same area we are getting A. It is not like A is here. Like if it is A is in different place and insertion is in different, then it would be two different mutation occurring in the same patient. But here, wherever there is insertion in that same sequence, we are getting a substitution also. That means it is a part of same mutation or variant.
So now, based on this, my reference sequence would be this GCC, GTTA, and my altered sequence would be in place of C, now there is insertion of GGGG, and now this C is being changed to A. So this is a delin that there are the change is happening in a consecutive nucleotides. So let's follow this. We go to ensemble, we check the exon, we see the cDNA, and we see our exact position where that change is happening. Now, if you see the Excel sheet that we have given and the Kava coordinates, we have put that instead of 11, we have put that the change is happening at 12. Can anyone tell me why this is it? Because here we can see there are two C's. So when you apply the 3' rule, the change is happening at this C and not this C. So the entire change, the GGGGA, I can say that this has happened at this C according to HGVS nomenclature. So the coordinates would be 897, 177, 12. So after applying 3' rule, now it becomes very much simpler to apply the HGVS nomenclature, that we just have to give a sequence identifier. This is my position. We have to add up delins, and whatever sequence that has been changed, we have to put ahead. So this is my genomic coordinate.
So why didn't you say 17712C delins, then what? ऐसा क्यों नहीं बोला? You directly said 17712 delins.
So it is not, the HGVS says that it is not necessary to mention what has been changed, but you have to mention what have been changed.
Because we know what is there in 17712, right? We have the sequence, we have the reference to us. So we know it's a C. So it will be like redundant information, so you do not need to give it.
Similar goes for transcript. At transcript 737, instead of this G, this change is occurring. So C dot 737 delins, we will write this sequence. Coming to protein, so for protein, HGVS says that unlike nucleic acid and protein coordinates are always prefixed with reference amino acid. So my original sequence was coding for protein position at number 246. But now after this change has, this GGGG is being added, it will result in a frame shift. This Gojiri has already shown that this will result in a frame shift, and after 8 amino acids, there will be a stop codon. So the frame shift change at 246 position will cause proline being replaced by arginine and will cause a protein chain termination at 8 amino acid sequence. So my protein variant would be P dot proline 246 arginine frame shift termination 8. HGVS says that it is not important to mention this number. You can just add an asterisk. So let's when we cross check this on variant validator, we will have similar results. Now coming to next case.
Okay. Skip this case for interest of time.
अभी तुम्हारा इसके cases बाकी है and lunch भी बाकी है और फिर workshop भी बाकी है. So everything will get derailed.
Yes. Last.
Just one thing I want to add in this case, that here also there is a complex change happening, deletion and insertion, and in between there is a single base. Now what HGVS nomenclature for delins says that these two nucleotides should be consecutive, but as we speak they are updating their criteria for delins. So they are saying that two variants that are separated by fewer than 2 intervening nucleotides, that is not including the variant themselves, should be described as a single delin variant. So this will, though there is a base in between, this will be a delin and not two separate mutations. And also if we apply 3' rule here, the original deletion was this and this was getting inserted after this G. But if you apply 3' rule real, so there is a G here and there is a G here. So the actual deletion would be GAGG and not GGAG. So then it becomes consecutive stretch of nucleotides. So rest of the things are similar. We add suffix delins and the change that is happening. Similar results we will get when we put on a variant validator. Thank you. Now I would like to invite Dr. Seema, who will be covering synonymous variants.
So Seema would be giving us an example of deletion that was involving both exon and intronic area. Please.
Good afternoon. So, since we are short of time and I have two cases to discuss, but I will quickly go through single case. So, this is a case of 40 years old male who was diagnosed with acute myeloid leukemia and cytogenetics was normal. So, this is NPO1 gene alteration. So, here we can see this whole complete 16 nucleotide sequence has been deleted and it is located on chromosome 5, and this is the start site at the level of genomic coordinate, and this is the end site. So, first we have to check whether it is a forward strand gene or the reverse strand gene. So, it is a forward strand gene. So, when I mapped the sequence, here we can see this is the start site of the genomic coordinate, which is where the change is occurring, and this is the end site of the genomic coordinate where the change is ending. And simultaneously, we can see that the change is happening throughout the exon 9. So, at the 3' region of the exon 9 as well as in the 5' region of the intron 9, that is intron between exon 9 and 10. So, this is 11 nucleotide sequence which is happening at the level of exon 9, and this is 5 nucleotide sequence which is deleted at the level of intron. So, total of 16 nucleotides. So, at the level of, so the HGVS description at the level of genomic coordinate will be G dot the coordinates, the start and the end site, and the deletion. Now coming to the coding, the description at the level of cDNA. So, okay. So, this is the coding DNA coordinate of exon 9. So, the exon 9 is beginning at this position, that is cDNA 670, and ending at this position 771. And our change is happening at this site, that is from coding DNA coordinate 761 to 771. And it is showing the 11 nucleotide sequence deletion has occurred. But since it is a coding DNA sequence, we will not be able to see the intronic sequence which is happening. So, so to make it simple, I have drawn this diagram, and here it is exons 9, and this is the deletion which is happening at the 3' region, and this is the start site of the deletion at the level of coding DNA, that is 761, and this is the end, and this is the end site of the coding DNA at the level of exon, not the intron. So, now here we can see there is 5 nucleotide deletion in the intronic region. So, how we will give the nomenclature. So, what will be the nomenclature? So, the nomenclature will be coding DNA 771, and since the change is happening 3' downstream of the exon, so it will be plus 5. So, just a hypothetical example, what if the change was in the 5' upstream of the exon? So, if there is 9 base pair, 9 nucleotide deletion, the nomenclature would have been C.670. So, it is coming from this. This is a 5' coordinate of exon 9 and minus 9, since the change is occurring in the intron 9 upstream of exon 5. Sorry, upstream of exon 9. So, the description at the level of cDNA. Okay. So, the description at the level of cDNA will be coding DNA 761 to 771, which is indicating the deletion happening at the level of exon, that is the 11 nucleotide deletion, plus 5, which is mentioning the deletion which is happening at the level of intron downstream of the exon, that is downstream of exon 9, that is 3' region, and deletion. So, now, So, now we will go through the description at the level of protein level. So, we see that in the intronic region, there is 5 nucleotide deletion, and this deletion is also spanning the canonical donor splice site, that is the GT. So, the splice occur at AG, which is a canonical acceptor splice site, and this is the donor canonical splice site. So, we really don't know that where the RNA spliceosome complex will splice subsequently. So, we really don't know. So, this is a image I will go quickly through. So, this is the image which I have taken from Wimmer K. et al. Article. So, here it is showing that if any alteration is happening at the 5' splicing site or 3' splice site or any branch point, that is 5' of the exon 2. So, it will skip this exon and it will lead to the juxtaposition of, it will lead to the fusion of exon 1 and exon 3. And what if there is a new splice site created? So, in our case also we don't know. So, whether it will skip the exon or a new splice site has created. So if I go back, there are many subsequent GTs. This GT, this GT, and this GT. There are so many GTs, so we really don't know which new splice site will be created. So if the new splice site is created, it will include exon 1 and this portion of the intron as well as this exon 3. So I think it's clear. I can move. Yeah. So at the level of protein, the description will be p dot query, because we don't know what is going to be the change until unless it is proven on function analysis. So this is the final HGVS variant.
Variant validator output. So usually when we get such query in variant validator, it is either involving the splicing area or it is involving the intronic region. That usually happens for this thing. So this is what we wanted to show with this case, which Seema has explained very well.
So I will skip the last case.
Yes, variant validator will, yes, this was just understanding of it. Otherwise you will get it in variant validator. In that, clinical significance will be covered tomorrow.
Thank you, everyone.
Thank God we survived. So now we will break for lunch. We assemble at 2:15 p.m. Not more than that, please. 2:15 p.m. at our respective workstation. We know who are in processing, who are in bioinformatics. And also today we have the party at Bahana, you must be knowing, at Belapur. So please join us there in the evening. But in the end, I would like to say that, when we decided that, I asked Shweta, how are you going to spend 3 hours on HGVS? But then I can only imagine how much effort it must have taken to put together all these examples. So fellows as well as Shweta, for so long, the examples are so varied, and to go through the routine reporting and actually pull them out. So thank you, Shweta, for that painstaking effort. And we'll break for lunch.
It's like, wait,
yes, we'll do that.
I
yes,
cannot do right now. I'll do it after.
Thank you. So the afternoon session and then party, please, everyone.